Right, to continue our discussion of cytology, study of chromosomes, we want to continue looking at autosomes and autosomal variation. And this is the largest number of chromosomes, obviously, that we would have. So we're talking about these 22 pairs of chromosomes that make up the majority of the genetic material that we have, and variation within those. Now, just to kind of pull us together is talk about things that can occur relative to humans. First off, once we move out of the sex chromosomes and move into the autosomes, we see far more severe multiple congenital anomalies, in other words, birth defects. We see a much larger collection of these in those either fetuses or those individuals that make it to live birth. The majority of live births are trisomies. Okay, these are so we're just adding an extra chromosome. Monosomy is essentially not tolerated, at least in human systems. The loss of a major autosome is usually a lethal system. Now, there have been, of course, abortices and some cases where monosomy has occurred. When it does actually occur in live birth, it's usually associated with mosaicism again, this idea that you've got some cell lines that are monosomy, but then you've got others that are normal diploidy. Now, very important, the next one is very important. Most of the cases we see of autosomal aneuploidy are what we would call completely de novo. There's no family history. If you go back into pedigrees, you don't see any evidence that there's been a case of this particular form of chromosomal problem occur. They simply occur just at random, and that's true for all the ones we're going to look at. Now, it says 95%, which means that there are examples, and we will come to those. We're going to actually have to go through a whole number of chromosomal variations until we talk about translocations, which account for this other 5%. And those are familial. Those tend to be passed down in families. But for the most part, there's simply de novo mistakes that occur. Now, because we're talking about autosomes, of course, we're talking about both genders, both sexes being affected. But that's usually not equal as far as making it to live birth. Remember, again, male and female fetuses have different expectations of actually making it. And when they have genetic effects, genetic anomalies, it even affects it even more. And we'll talk about a couple of cases where you see extreme variation in the amount of impact that the sex of the individual has. All right, so we're going to start with the first one, and it's it's listed as the most common genetic syndrome in the world. I think probably Klinefelter's is more common, but certainly of the autosomal, there's no question that these individuals express the most common syndrome that occurs that we see. Okay, and this is, of course, Down syndrome. Down syndrome is a trisomy of the 21st, meaning that you'll have 47 chromosomes. This is an ideogram showing an individual where you have this 21st. Now, just take a look and think for a moment. These are very small chromosomes. They're certainly, they're about a fifth of the size of some of our largest chromosomes and a third of the size, even things that are nearby. So we're not talking about huge amounts of DNA. We're not talking about losses of DNA. We're simply talking about the fact that you've gained an extra chromosome. So instead of having two homologs that you would normally have, one parent contributed two. So you end up with three. Okay, and that's important because as we work through this, we're going to walk up to some of the larger ones and show you the effects. All right. Okay, now what do we have here? Well, what we get from Downs, again, it's going to be a syndrome, so the variation will be dramatic as to what the components are, what the phenotypic expressions are. But this beautiful young lady right here represents some of the classic things that you might see, which are including a flat facial profile. The shape of the face will be there. The eyes will tend to be pointed more up. Sometimes there's a gaping mouth, but not always, and it may have a protruding tongue at some points. So they have certainly this flat facial profile is something that you will very often see and very recognizable, and certainly the facial structure is something that's very common that you'll see in Downs individuals. Okay, so some of the things that do affect it, they do often have delayed motor development. They will have these epicampal folds, these eye folds that tend to make the eyes look like they point up. They're individuals that will rapidly age. In fact, there's a high, once they reach their 20s and 30s, there's actually a high incidence of Alzheimer's or Alzheimer's-like uh, dementia. It doesn't necessarily have to be Alzheimer's, but, and the idea is because there are some proteins related to amyloid beta that are apparently associated on the 21st chromosome. So you get an overproduction of these amyloid betas. And what happens is when this happens in neural cells, this is what happens with things like Alzheimer's, those proteins can't be processed effectively and they're simply stuck on the outside of the cell. If it's a neural cell, eventually you get so much of that material out there that you can't fire properly, and that's one of the causes of Alzheimer's is that these proteins are put on the outside, and these individuals apparently make an extra large amount of this protein. They tend to have heart trouble, and those individuals that do have valve troubles and other issues are often the ones that have the least likely long-term life expectancy. Those individuals who were born without these or who these are paired can be extended quite a bit. They have what's known as a Simian crease. It's known as that's a pejorative term, unfortunately, for it because it relates to the great apes, the simians. But in the great apes, instead of having lines that go across like at an angle in our lifelines, they go across. And this picture here is a good example of these creases. The more appropriate term, the more term used in medical systems would be a single transverse planar crease. But if you look it up, you'll see it called simian because the simians, the great apes grab like this, and of course we grab more like this. But unfortunately, it's the unfortunate name for it. But that's very common. It's also seen in lots of other things. Fetal alcohol syndrome. Children will have these types of creases. You'll see them in a number of systems where you get some disturbed development during your intrauterine growth. As I said, they tend to have this flat facial profile. They can have undeveloped secondary sex characteristics. They're often diabetic. There's some leukemias that are common in these individuals. Although interestingly, there's apparently there's an overproduction of some protective proteins on the 21st. So solid tumors are not as common, although their blood tumors and some of the other types of cancers are more common. Life expectancy, a major system we see here. In 1960s and before then, the life expectancy was extremely low. It had increased dramatically after World War II because of the introduction and widespread use of antibiotics, because a lot of the issues they would have would simply be from bacterial infections and from other types of infections because of their immune system. So there was a spike, kind of an increase then. By the 1960s, it still hasn't increased much, improved, and it started to improve by the 1980s. What really drove some of this was movement of these individuals out of institutions. They had been, for the most part, in the last century, had basically been pretty much institutionalized and under fairly severe conditions, and the life expectancy there was pretty bad. As the medical conditions started to improve, by 2007, I think the life expectancy was somewhere around 47 years. Currently, the most recent estimate I saw was greater than 60 years. All due to medical care, and so that's a massive increase due to the conditions in which these individuals were placed for living, as well as medical care. Now, unfortunately, the National Institutes of Health, which is one of the major areas in this country that supports research toward these types of things, funding for Down syndrome actually decreased dramatically there over the last decade, while others, of course, Alzheimer's and things increased. But even with that, you see a dramatic increase in the survivorship of these individuals. Now, again, a majority of these trisomies are lost as miscarriages. There is a lot of loss during the first year of life, but if you can make it past that with medical care, they actually can move forward and have long life expectancies. There's urban myths that they're non-reproductive, and that's generally true. It's particularly true of the males. I think in the U.S. only at this point about 3 or 4 males have been formally recorded as being fathering children that were Down individuals. and it's usually sperm production. Apparently, there's the sperm production is unsuccessful, but there have been a number of females that have produced somewhere in the 20s, high 20s, of well documented cases. Unfortunately, some of those occurred, in situations with institutionalized individuals. Other times they haven't, but a lot of the early ones were actually, in conditions where reproduction wasn't being controlled. They have about a 50-50 chance of having, if it is a Down's female, of having an unaffected child. You can think about the process of meiotic division to see why that might be true, and it is true. Okay, now it's probably not, even though the range of capabilities of Down's individuals is huge, probably having a Down's female reproduce might not be the best thing, but that's a family choice, not, one thing that's really important about Downs is that it was the first of these genetic issues that was related to what's called maternal age effect. And this occurred back, Downs was actually described in 1866 originally, and then 1883, another name. So that's why Downs is named for the first person to describe the condition. They didn't know it was chromosomal. They just knew that they had this collection of syndromes. But starting in the 1920s, a couple in the UK that the Penroses that worked in some of these. He was a physician and she was well, and they worked in these institutions, and he started to notice that there was a relationship between age. Penrose is very famous. He was a just amazing mathematician as well as physician, and the Penrose stairs is one of his designs. He was a puzzle maker, so he drew these Penrose stairs. Have you seen the Matrix or something where they're walking down and these people keep coming up, passing them? They're in a Penrose square, illustration of that. But Penrose did many things. But one of the things he and his wife worked on were the instances of Downs among females. And being a statistician and mathematician, what he noticed was that there was a significant increase in the occurrence of that, and actually came up with this sort of model where today we would say that if you're 19 and we give you a 1x chance of having a Down, say 1 in 2,000 chance or whatever it might be, if that same female reproduces at age 30, she's going to have about 3 times the chance of having a Down's individual. Yeah. If she waits until she's in her 40s, that will increase by 20x. And if she goes, if she reproduces past 45, it dramatically increases to 50x. So the females that are over here in this region really have a high incidence of, it turns out not just to be Downs, but many chromosomal aberrancies that are related to aneuploidies actually dramatically increase as women reach and reproduce in their 40s. And this has had some pretty dramatic effects around the world. You see the age of last reproduction in developed countries tends to fall. Women are reproducing less and less, and they're, as they can control in places that they can actually control their reproductive output. You also see that this was a pattern that became important in many developed countries in recognition of the fact that the effects on women over time are greater than on men in the workplace. And so OSHA rules and things, particularly our lab used to use very small amounts of radioactivity. My body burden at any one time, my amount could be much higher than females working in a lab. Now, one of that was because I was physically larger than some of them, but even in general, the things that they can be exposed to, the rules about what women can be exposed to as men, as far as the conditions, can be affected, and were codified and changed by these sorts of observations that as women increase. Why is that? Well, you think about it, males produce sperm constantly. Sperm has a life cycle of about 21 days in humans, and we're reproducing it consistently. If it's not used completely by 21 days, it's reabsorbed and they're refunctioned, but we have sperm production going on continually through that time. So as we reach 40 or 50 years old, the sperm that we're actually using technically is only a few weeks old. In females, the development of these cells that are going to be the primary oocytes, or the meiotic cells, are arrested in the ovaries. Okay, and those cells are there shortly before birth until shortly after birth. The process continues, and they sit there for the life of that female as long as she's ovulating. And about every 28 days, one or more of those cells will complete the meiotic process. The oogenesis process will be excreted from the ovary and will be ovulated. Okay, so point of that is these cells that are arrested in meiosis, that are going to become the eggs, have been sitting here for 45 years. They've been sitting here 20 years. So any insults to that ovarian system could result in final problems with the formation of the spindle fibers and all sorts of things that can occur. So this is the primary difference that we see here is just the longevity, the amount of time that the gamete has actually been there, the progenitor egg cells versus the sperm cells. Okay. Now it turns out it's not quite that simple. There also turns out to be paternal age effects. As males age, we have problems. One of the classics is that the de novo mutation rate goes up in males. The older males tend to be those associated with autism and schizophrenia and other things. So as males increase, not only even though we replace the sperm, the process of meiosis, there's more and more error rates that occur over time. And this doesn't actually happen in females. It's interesting. Now, there's more chromosomal issues, right? But as far as de novo point mutational systems, females tend to flatten out, males tend to increase. And you can imagine why, because the replication is one of the places where mutation rates are high. Well, females have already done all the replication. Remember, I keep arguing over and over that replication is off down here somewhere in time, and meiosis is way off up here. Well, the males have to keep going through that cycle, but the females don't. They simply complete the process of maturing the egg and the process of meiosis, and so there's no replication errors. There can be other errors that occur, but the rates actually kind of fall. So point of that is there are later age females, it is important that they consider the fact that they could have chromosomal issues, but it's not just the women, it's also the men that can have these, both in terms of point mutations as well as it turns out now chromosomal mutations actually increase. So I guess the point is you probably want to reproduce when you're young, okay, and maybe if you're going to try to do that. Okay, all right, so that's looking at the 21st. All of that is situated with these small chromosomes down here. The 22nds, if you have trisomies of those, are generally lethal, as well as 20th. But two others that tend to show up. Now, if you go through Buck Gonker's book, okay, or his websites, you'll see that there are examples for all of these chromosomes that are out there, and very interesting issues of aneuploidies for these. But we're going to concentrate on just two, and the reason we're just going to concentrate on them is just to show you, even moving up a small amount of DNA and imbalancing the system just a little bit causes just a massive increase in the outcomes. So we had these small chromosomes right here. We're going to slightly more than double them, maybe 2.5 times over here on the 18th, and then we're going to double that over here on the 13th, okay, and see what the effects of those are. These are some of the really common chromosomal aberrancies that we see. The trisomy of the 18th is known as Edwards syndrome. And this is just a trisomy again. So you're going to end up with normally having 2 18ths. Now we've got 3. Okay, I showed you those on there. These individuals tend to have an elongated skull. They often have what are known as skin redundancy. So there's the skin can be pulled or moved in sort of layers. Heart defects, webbed neck. They frequently have a horseshoe kidney. What that means is nephratic tissue that's supposed to form the medulla and other parts of the kidney doesn't complete. So you've got this, the cell lines are there, but they don't actually form completely. So these individuals either don't form a full kidney, excuse me, or they have a dysfunctional kidney if it does form. They have severe cognitive disabilities. So their thought process, and then again, this is probably due to some of this interuterine imbalance as they develop, and the neural system being heavily affected by that. Okay? The occurrence, and I said that just because both sexes can be affected, obviously because they're autosomes, doesn't necessarily mean they make it to live birth. There's about a 4-to-1 ratio of males of females to males actually at birth and surviving this. So males that have Edwards syndrome tend to not make it through the gestation process. The vast majority of these individuals die within the first 6 months. That's when There's only about 10% make it the first year, even with good medical care. That's at least the last data that I have. There are females now making it into their 20s and 30s, which was pretty much unheard of, say 20, 30 years ago. But they are making it. They do require, great amount of care, full attention for them pretty much that whole time. But they are, at least the individuals are making it. And again, it's females. Males rarely make it past the first year. I know one person that had Edward's child personally, and they made it to almost their fifth birthday. So that was for them a very important milestone that the child made it to. But for the most part, males, and that was a male, they don't tend to make it very long. Okay. Now, just moving up the scale, all right, so we've little over doubled the amount from 21st to 18th. Now we're going to jump over to the 13th, which is another one that's seen as a live birth, okay? This is a trisomy of the 13th. It's known as Patel Syndrome. It has pretty much many of the same issues that Edwards has, as far as heart conditions, these other things, but we get many, many more severe issues going on. They tend to have motor seizures, which means that they will shake or move until they go into tetany, and they'll have, then they'll stop and they'll go back into tetany by shaking or having these motor seizures. The cleft palate, which also relates to a bilateral hair lip, so the top of the palate doesn't form properly. Actually, this goes all the way into the neural system, because the major parts of the brain, our lobes of our brain separate into two parts. They often don't do that. With individuals that have Patel syndrome, they don't get this proper creasing. They tend to have things like necrotic scalp ulcers, these opening spaces that will occur and are susceptible to infection and all sorts of things. They have post-axil polydacty, that's kind of shown here. These forms. So post-axil generally is extra toes or extra fingers that come up apparently. Many are premature if they do make it, if they're not stillborn or they do make it. And then the mortality rate is extremely high. About 50% are lost within a couple of weeks after parturition, after birth. They rarely live, if they make that, they rarely live past 5 years. And right now, maybe 10 years is really pushing the extreme on these with really, really good care. And as you can see for this young man right here, they tend to age very rapidly. So if they do make it, past a few years, they start to age significantly and loss. So the vast majority. So the point of this is that it's a very severe effect, right? For the most part, these are not going to be functional humans in the sense of being just able to take care of themselves and be, in a societal sense. And all we've done is gone up to the smallest. We haven't gone to the 7th or 8th or 9th or 10th. All those exist. There are births that have occurred from these. Certainly there's been vortices that are documented, well documented with these. They occur, they just don't make it. Okay, so once you get past the 13th and start moving forward into the number, you really, the rarity of these goes sky high. You don't see these coming to live birth very often at all. All right, well, so we've looked at aneuploidy, and we've gone through all of these now. We looked at the sex chromosomes. We talked about Turner's and Klinefelters and how those tend to be related to each other. We talked about multiple X syndromes. We talked about multiple Ys. So we have XYY as well as XXYY syndromes that we can see that are the sex chromosomes. And in general, these are fairly, they can range. They're important. Unfortunately, they do cause a lot of miscarriages when they occur, so there is a lot of loss of these fetuses. But once they make it to live birth, there are individuals that can be quite normal individuals in full sense of it. Then we have autosomal ones, and we just gave you an example of 3. There are many, many we could go through, but Downs being by far the most common form that's out there, and the range of that again can be from very, very functional, highly functioning individuals to individuals who are much less able to function. But then as you move up the scale, you rapidly start to have major issues, and the life expectancies, the outcomes of these, the amount of clinical care required just escalates highly, going up even to small amounts of this DNA. Okay. Again, aneuploids here. Now we could talk about aneuploids in horses and cattle and chickens and all sorts of things. But these are good examples, I think, bring home for humans what the effects of these can be. All right, that ends our discussion about changes in chromosome number. The next series of lectures on this on cytology, we're going to deal with chromosome structure. So we're not going to lose or gain entire chromosomes, but we may lose or gain pieces of chromosomes, or we may move pieces of chromosomes around as we talk about changes in structure.